Hi, hello. Good morning. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College, Autonomous, Mehboob Nagar. In our last class, we saw about photobiology and photophosphorylation, that is cyclic and non-cyclic photophosphorylation. And we saw how ATP and NADPH are formed in light reaction. We said that the light reaction takes place in the grana of chloroplast, and as a result of the light reaction, the assimilatory power generates, that is ATP and NADPH. So today we will see the dark phase of photosynthesis. Okay. Dark phase is the second phase of photosynthesis. It involves reduction of carbon dioxide, for which light is not required. It is a misnomer. Dark phase that does not represent that it takes place in dark, but the light directly is not involved during this phase, so it is named as dark phase. Okay. uh, it was first discovered by Blackman, and the reducing power, that is ATP and NADPH, produced in the light phase are utilized to reduce carbon dioxide. And the sequence of reactions involving carbon dioxide fixation and formation of complex carbohydrates is called the path of carbon in photosynthesis, and it can be traced by using the techniques such as radioactive carbon technique and paper chromatography. We will see what is the radioactive carbon technique and paper chromatography. Radioactive carbon technique is nothing but by making use of carbon 14, a radioisotope, uh, is used in tracing the intermediate compounds formed during the assimilation of carbon dioxide in photosynthesis. C14 atoms behave in precisely the same manner as a non-radioactive carbon. They enter all the reaction sequences and label the compounds that are involved in the path of carbon. The labeled or tagged intermediate compounds can be identified and assayed. This is the radioactive carbon technique. Sorry. Then paper chromatography technique. Paper chromatography technique is a technique of separating the photosynthetic compounds from a complex mixture of organic compounds. In this, the plant tissue is extracted with a solvent. A few drops of the extract are applied to a corner of the filter paper. It is then placed in an organic solvent. The solvent moves over the extract by capillary action and separates the different constituents according to their relative solubility in the solvent. And when the chromatogram is examined with a Geiger-Muller counter, the compounds containing radioactive C14 are detected. Labeled compounds are detected by autoradiography. In this technique, the chromatogram is exposed to an X-ray film. C14 containing compounds will produce an image on the film, while the unlabeled compounds will not produce any image. So this is how the intermediate compounds are uh, identified in the carbon pathway. So these techniques were used by Kelvin, Benson, and Basham and associates to separate, isolate, and identify the organic compounds formed during carbon dioxide fixation. And they used chlorella and centesmus as experimental material. Okay. So Kelvin and Benson were able to establish the path of the carbon assimilation and is named as Kelvin cycle, photosynthetic carbon reduction cycle. Uh, and For this, Kelvin was awarded Nobel Prize in 1961 for this significant work. Plants in which CO2 is initially fixed as 3-phosphoglyceric acid are known as C3 plants because the first stable compound formed is a 3-carbon compound, so we call them as C3 plants. Kelvin cycle occurs in the stroma of a chloroplast. It was observed that um, when radioactive carbon dioxide was supplied in the Dock reaction, PGA increased, but there was a decrease in the concentration of RUBP, that is ribulose bisphosphate. This shows that ribulose bisphosphate is the carbon dioxide acceptor during the formation of phosphoglyceric acid. And some of the PGA is converted to sugar, but the balance is utilized for regeneration of RUBP. The cyclic pathway that uses phosphoglyceric acid to form other sugar phosphates and regeneration of RUBP is collectively known as Kelvin cycle. The Kelvin cycle consists of three main parts. What are the three main parts? Carboxylation, reduction, and regeneration. Okay, the uh, a molecule of ribulose bisphosphate is carboxylated by CO2. Carboxylation is nothing but entry of carbon dioxide, hmm, is the fixation. Carboxylation is nothing but a fixation of carbon dioxide into a stable organic intermediate where CO2 is utilized for the carboxylation of RUBP. And an unstable 6-carbon compound is formed which splits into two molecules of phosphoglyceric acid. This reaction is regulated by an enzyme called ribulose bisphosphate carboxylase oxygenase. Ribulose bisphosphate carboxylase oxygenase. See here, RUBP is the primary acceptor of carbon dioxide, and this reaction takes place in the presence of RUBP carboxylase oxygenase. It is popularly known as Rubisco, and this enzyme is the most abundant protein in the world. It is the key enzyme in biomass production from carbon dioxide in plants. It catalyzes the insertion of carbon dioxide and simultaneous cleavage of ribulose bisphosphate into two molecules of phosphoglyceric acid. So the first formed stable compound is a 3-carbon phosphoglyceric acid. So these plants are also known as C3 plants, and this cycle is also known as C3 cycle, Kelvin cycle, or C3 cycle, or reductive pentose phosphate pathway. And uh, by the formation of this PGA, carboxylation process is over. Then comes a reduction, very important reduction. Here, the 3-phosphoglyceric acid is reduced to 3-phosphoglyceraldehyde. See, this is the glyceric acid. This glyceric acid is reduced to glyceraldehyde. But before changing into aldehyde, uh, it first undergoes phosphorylation. It takes in the presence of ATP and phosphoglycerokinase. This PGA, phosphoglyceric acid, it forms 1,3-bisphosphoglyceric acid. Okay, that is phosphorylation of PGA takes place before its reduction. Okay, here ATP are utilized. Uh, See, for 6 carbon dioxides, 6 RUBP are utilized. As a result, 12 PGA are formed. For each PGA, 1 ATP. So a total of 12 ATP are utilized for phosphorylating this PGA. Resulting product is 1,3-bisphosphoglyceric acid. Okay. So PGA is phosphorylated by ATP formed in photophosphorylation. It results in the formation of 1,3-bisphosphoglyceric acid and is catalyzed by phosphoglycerokinase. In the next step, this 1,3-bisphosphoglyceric acid is reduced to 3-phosphoglyceraldehyde by NADPH. This reaction is catalyzed by triose phosphate dehydrogenase. See, you can see here this 1,3-bisphosphoglyceric acid. It undergoes reduction. It makes use of the reducing power, that is NADPH, which gets oxidized to NADP, and the 1,3-bisphosphoglyceric acid is converted to glyceroldehyde 3-phosphate. So this is the reduction reaction. So carboxylation and reduction. The remaining is a regeneration. Regeneration of what? Regeneration of ribulose bisphosphate, which was first utilized to fix the carbon dioxide. See, this ribulose bisphosphate has to be regenerated to continue this cycle. So here we have uh, a total of 12 molecules of glyceroldehyde 3-phosphate. So out of these 12 molecules, 6 molecules are isomerized into dihydroxyacetone phosphate. Here, for our convenience, in this cycle, we have shown only uh, 6 molecules of glyceroldehyde 3-phosphate, but here at every place, instead of 1, you have to imagine 2. So as a whole, 12 PGR, PGEAL glyceroldehyde 3-phosphate are present. Okay, so here out of 12, 6 they turn into DHAP, dihydroxyacetone phosphate. They isomerize. Okay, so mm, DHAP is also a triose phosphate. This isomerization takes place in the presence of the enzyme called triose phosphate isomerase. One molecule of glyceroldehyde 3-phosphate and one molecule of DHAP condense, forming one molecule of fructose 1,6 bisphosphate in the presence of enzyme called aldolase. See here, one molecule of DHAP and one molecule of glyceroldehyde 3-phosphate. They condense in the presence of enzyme called aldolase and forms fructose 1,6 bisphosphate. And this fructose 1,6 bisphosphate becomes fructose 6-phosphate by removal of a phosphate molecule from it. See, inorganic phosphate is removed from this one, and fructose 1,6 bisphosphate turns into fructose 6-phosphate in the presence of an enzyme called phosphatase. Okay, then one molecule of this fructose 6-phosphate, it condenses with one molecule of glyceroldehyde 3-phosphate in the presence of the enzyme called transketolase. Here it is a 6-carbon compound, and this one glyceroldehyde 3-phosphate is a 3-carbon compound. So as a whole, 9 carbons are taking part in the reaction. So these 9 carbons are split into one 5-carbon compound called xylulose 5-phosphate and one 4-carbon compound called erythrose 4-phosphate. So this reaction takes place in the presence of the enzyme called transketolase. So here we have this xylulose 5-phosphate. It undergoes epimerization reaction and produces ribulose 5-phosphate. And this erythrose 4-phosphate, it again condenses with one more DHAP, that is a dihydroxyacetone phosphate, which is an isomer of glyceroldehyde 3-phosphate in the presence of an enzyme called aldolase. So this 4 carbons plus this 3 carbons, a total of 7 carbon compound is formed, which is called as pseudoheptulose 1,7 bisphosphate. So this reaction also takes place in the presence of the enzyme called aldolase, and immediately this pseudoheptulose 1,7 bisphosphate loses one phosphate ion in the presence of a phosphatase enzyme, and the resultant structure formed is pseudoheptulose 7 phosphate, which is a 7 carbon compound. And this pseudoheptulose 7 phosphate, it condenses with one more the remaining glyceroldehyde 3-phosphate, the 7-carbon compound plus 3-carbon compound, total of 10 carbons split into one xylulose 5-phosphate and one ribose 5-phosphate. So we have a total of 2 xylulose 5-phosphates and one ribose 5-phosphate at the end. So these xylulose 5-phosphate, they undergo epimerization reaction resulting in the formation of a ribulose 5-phosphate, and this ribose 5-phosphate, it undergoes isomerization resulting in the formation of ribulose 5-phosphate. Here this ribulose 5-phosphate, it undergoes phosphorylation reaction because the initial product which we used during carbon dioxide fixation is ribulose 1,1,5 bisphosphate. So this ribulose 5-phosphate, it undergoes phosphorylation. That means it takes up 6 ATP molecules, and 6 ribulose 1,5 bisphosphates are regenerated. So this process, regeneration process, is also over, and a formation of hexose. Here, hexose formation and sucrose formation occur in a cytosol since uh, glyceraldehyde 3-phosphate are transported out of chloroplast via antipode carrier system, and glyceraldehyde 3-phosphate and dihydroxyacetone phosphate condense under the influence of enzyme aldolase, and forming fructose 1,6 bisphosphate. And removal of phosphate by enzyme phosphatase results in fructose 6-phosphate. Later on, this fructose 6-phosphate is converted to glucose 6-phosphate. So this glucose is formation of glucose takes place in the cytosol, that is cytoplasm. Okay, so this is how fixation carbon dioxide is fixed into a glucose molecule. See, we for each glucose has a 6 carbons, so uh, we need 6 carbon dioxides for formation of a single glucose molecule. So 6 carbon dioxide molecules, they um, the primary acceptor is a ribulose 5 1,5 bisphosphate. So 6 molecules of a ribulose 1,5 bisphosphate, they accept 6 carbon dioxides, resulting in the formation of a 12 phosphoglyceric acid, which is a 3 carbon compound, and this process is known as a carboxylation process. Later on, it undergoes reduction reaction. So this phosphoglyceric acid, which was not ready to undergo any further reactions, it first takes up phosphate ions from ATP, and 1,3 bisphosphoglyceric acid is formed. This 1,3 bisphosphoglyceric acid, it undergoes reduction making use of 12 NADPH. So as a result, 12 uh, 12 molecules of uh, glyceraldehyde 3-phosphate is formed. So we have total of 12 glyceraldehyde 3-phosphate. So one glucose requires 6 carbon atoms. So out of 12 3 carbon compounds, we require only 2 3 carbon compounds to produce one glucose molecule, and the remaining 10, they regenerate the ribulose 1,5 bisphosphate. So this is known as a Kelvin cycle. Here a total of 18 ATP are utilized and 12 NADPH are utilized for formation of one glucose molecule. This is Kelvin cycle, and uh, until 1965, it was thought that all the green plants fix carbon dioxide through this Kelvin cycle. But in 1965, Korchak, Hart, and Burr found that when sugarcane leaves fed with radioactive carbon dioxide, most of the labeled carbon appeared in malic acid and aspartic acid. These results were confirmed later by Hatch and Slack in 1965. They discovered that radioactive carbon dioxide is fixed as oxaloacetic acid, which is the first intermediate product of a carboxylation. And the primary acceptor of carbon dioxide in these plants is phosphoenol pyruvic acid, that is PEP, whereas in C3 plants, the primary acceptor is RUBP. Okay, so um, since the Products of CO2 fixation are four carbon compounds like oxaloacetic acid, malic acid, aspartic acid. The plants exhibiting this pathway are called C4 plants. Hmm. Um, as Hatch and Slack identified its pathway, it is also known as a Hatch and Slack cycle. And this Hatch and Slack cycle operates in subtropical and tropical grasses and in some dicotyledons too. Okay, a total of uh, uh, 117 genera of plants belonging to 13 families having C4 mechanism of CO2 fixation, and it occurs in certain grasses like Digitaria, chloris, saccharum, sorghum, ziames, amaranthus, tribulus, etc. Okay. Mm. In Hatch and Slack cycle, the primary acceptor of carbon dioxide is phosphoenol pyruvic acid and not ribulose bisphosphate, which is the CO2 acceptor in C3 plants. Hatch Slack operates in the mesophyll cells. It is also known as C4 cycle or dicarboxylic acid cycle or the beta carboxylation pathway. In the first step, see here, in the first step, what happened? Carbon dioxide reacts with a water molecule and forms a bicarbonic acid. See, H plus ion and bicarbonate ion. Hmm? It's CO3 minus ion. And this HCO3 minus ion reacts with phosphoenol pyruvate in the presence of enzyme phosphoenol pyruvate carboxylase, forming oxaloacetate. This oxaloacetic acid is a dicarboxylic acid. So the cycle, the first formed compound is a dicarboxylic acid. Uh, so this cycle is also known as dicarboxylic acid cycle, and the first formed stable compound has four carbon atoms. So it is also known as C4 cycle. Okay, and it's also known as one more name for this C4 cycle is beta carboxylation pathway. Okay, the oxaloacetate is readily converted into malic acid or aspartic acid depending upon the species. Okay, so this oxaloacetic acid, it undergoes reduction in the presence of NADPH and the enzyme malic dehydrogenase. Malic acid formation takes place. This malic acid enters into bundle sheath cells. Okay, so initially all these reactions are taking place in mesophyll cells, but uh, the after the formation of this malic acid enters into bundle sheath cells where it undergoes oxidative decarboxylation in the presence of NADP malic decarboxylase enzyme. Here, NADP uh, receives the H plus ions from this malic acid, and the utilized NADPH is again formed here. Okay, so it is an oxidative decarboxylation reaction. So the carbon dioxide is released here. Okay, and the H plus ions are also released. These H plus ions are taken by this NADP and reduced to NADPH, and carbon dioxide released, it enters into the C3 cycle, and the a whole C3 cycle again occurs here in the bundle sheath cells. Okay, so the malic acid, after undergoing oxidative decarboxylation, it forms pyruvic acid, and this pyruvic acid enters into mesophyll cells again, and the mesophyll cells, in mesophyll cells, this pyruvic acid, it takes ATP molecules, and in the presence of pyruvate kinase, again the phosphoenol pyruvic acid is regenerated. So here the regeneration of PEP also completed, and the carbon dioxide, which is released as a process of oxidative decarboxylation of malic acid, it enters into the C3 cycle, and carbon dioxide fixation takes place again in the same manner as it occurred in C3 cycle. Okay, so here one thing uh, to note is that in C4 plants, leaf anatomy are Cran's anatomy is seen. C4 plants have a specialized leaf anatomy called the Cran's type. The vascular bundles are surrounded by a layer of parenchyma cells known as bundle sheath cells that separate them from mesophyll cells. The mesophyll cells and the bundle sheath cells are in close contact with one another. They are connected by plasmodesmata. Therefore, all substances moving between uh, leaf mesophyll cells and leaf vascular bundles must necessarily pass through bundle sheath cells. The bundle sheath cells of many C4 plants contain more chloroplasts, mitochondria, and other organelles. And on the other hand, in C3 plants, bundle sheath is not distinct, and even if present, the sheath cells contain few organelles than the mesophyll cells are no organelles at all. In bundle sheath cells of maize and sugarcane, chloroplasts have no grana or rudimentary grana, in contrast to mesophyll cells, in which chloroplasts have well developed grana. And enzymes of Hatch Slack cycle occur in mesophyll cells, while those of Kelvin cycle are present in bundle sheath cells. The chloroplast dimorphism cannot be uh, taken as a rule, since in Bermuda grass, a well-developed grana are seen in chloroplast of the bundle sheath cells. So this is how the C3 cycle and C4 cycle, the fixation of carbon dioxide into glucose, takes place. Now we will see the differences between, first of all, we will see the significance of C4 cycle. The significance it has is adaptation to dry, hot environment, higher rates of photosynthesis due to CO2 affinity for phosphoenol pyruvic carboxylase, which fixes CO2 more efficiently even at very low concentrations. It prevents the escape of respiration CO2 from inner tissues. Photorespiration is undetectable, which results in greater photosynthesis. C4 plants show higher rates of translocation of photosynthetic products and efficient uses of water. So this is the significance of Hatch-Slack cycle, and we will see the differences between C3 and C4 cycles. So C3 cycle seen in subtropical and temperate plants, C4 cycle seen in tropical grasses and dicotyledons of Gramineae, Amaranthus, Cyperaceae, Portulaceae. Okay, in C3 plants, uh, C3 plants, Cran's anatomy is not seen, but in C4 plants, Cran's anatomy is seen. Bundle sheath cells have less chloroplasts here. There, they show number of chloroplasts. Chloroplast dimorphism is not seen in C3 plants, whereas chloroplast dimorphism is seen in C4 plants. The primary acceptor of CO2 is RUBP in C3 plants, whereas the primary acceptor of carbon dioxide is phosphoenol pyruvic acid. In bundle sheath cells, it is RUBP again. Okay, so the first stable product formed is PGA, phosphoglyceric acid, which is a 3-carbon compound. And in C4 plant, the first stable product formed is oxaloacetic acid, which is a dicarboxylic acid and a 4-carbon compound. And the optimum temperature is 10 to 25 degrees centigrade for C3 plants, and for C4 plants, it is 30 to 45 degrees centigrade. And in C3 plants, only C3 cycle occurs, but in C4 plants, both the C3 and C4 cycles occur. And photorespiration is present in C3 plants. Photorespiration is absent in C4 plants. CO2 compensation point is 50 to 150 ppm. CO2 compensation point in C4 plants is 0 to 10 ppm. To fix one molecule of carbon dioxide, 3 ATP are required, and in C4 plants, to fix one molecule of carbon dioxide, a total of 5 ATP are required. Here, oxygen inhibits photosynthesis, and in C4 plants, the presence of oxygen has no effect. So these are the differences between C3 and C4 plants. So this is our today's class.